Hi there, my name is Sydney Dukes, and I will be giving a presentation today on the use of RT-PCR testing to detect the COVID-19 virus. So COVID-19 is a contagious infection that is caused by the SARS-CoV-2 virus. This virus attacks cells in the throat, nose, and lungs. According to the CDC, COVID-19 is transmitted by respiratory fluids that are carrying the virus and can be spread through coughs or sneezes. Once an individual comes in contact with the virus, it will enter into cells in their throat, nose, or lungs. The virus then overtakes the cell's mechanisms and begins creating new viruses to further the spread of the COVID-19 infection. So the principle behind RT-PCR techniques. So RT-PCR is short for reverse transcription polymerase chain reaction and is a technique used to test for COVID-19. The virus that causes COVID-19 contains genetic material that carries instructions the virus uses to replicate the virus to cause further infection. RT-PCR detects the presence of a portion of genetic material within the COVID-19 virus. It does this by using DNA polymerase to create more genetic material within a sample, including the genetic material from the COVID-19 virus. Creating more genetic material in multiple cycles allows for the presence of the COVID-19 virus to be detected. So the viral targets used in detection of an active infection. So the COVID-19 virus can be detected by genetic material that's called the N gene, which holds instructions to create part of the inner shell of the virus. The RT-PCR process creates more copies of the N gene so that the virus can be detected within the sample. So the genetic material of a sample is monitored over time. The sample is then evaluated with a fluorescent probe, which quantifies the amount of genetic material from the COVID-19 virus after each cycle. If genetic material is present, the sample is positive and therefore it contains the COVID-19 virus. If there is no genetic material detected, the sample is negative and therefore does not contain the COVID-19 virus. So the RT-PCR testing is able to detect the COVID-19 virus in a sample. The presence of the virus indicates that the individual that the sample was collected from has the COVID-19 virus and therefore is considered to have an active infection. An active infection means that an individual has COVID-19 and is therefore able to transmit the virus, whether or not they are symptomatic from the virus. RT-PCR testing results are not influenced by past infections. There are different tests available, such as antibody or serology tests that will test for the evidence of past COVID-19 viral infections. If you or someone you know has tested positive for COVID-19, please isolate for at least 14 days and ensure that a negative test result is obtained before ending the isolation protocol. For this presentation, I have referenced information obtained from the Centers for Disease Control and Prevention, University of Pennsylvania Health System, DNA Learning Center, and Nucleus Medical Media. Thank you.